So, hello everyone. It's a big pleasure for me to be here again, to see you, either here in the room or online. And I will make a summary of our collaborative project in which 4 groups, 2 from Barcelona, Silvia Ginés and Guadalupe Mengot, and 2 from Madrid, Javier Fernández-Ruiz and our lab, have been working together in this couple of years trying to characterize a new interacting protein, GRP78, of cannabinoid CB1 receptor. As you most likely know, CB1 receptor is the main target of cannabinoids. CB1 receptor can be engaged and activated by either exogenous cannabinoids, essentially THC, the main bioactive component of cannabis, or by endogenously synthesized cannabinoids, the so-called endocannabinoids, the eicosanoids and andamide and 2-AG. CB1 is one of the most abundant metabotropic receptors in the mammalian brain, where it mediates a lot of effects, for example, cognition, memory, and learning, food intake, appetite, metabolism, movement control and coordination, fear, anxiety, nausea and vomiting, etc. In the context of CIBERNED, it's of course worth mentioning that there are many reports showing that cannabinoids induce neuroprotection in a large variety of animal models of brain damage and neurodegeneration. These studies so far are only preclinical, have been conducted only with animal models, especially mice, but also rats and pigs in some instances. But so far, the clinical efficacy of cannabinoid has been shown for the palliation of symptoms in patients with some neurodegenerative diseases. For example, inhibition of spasticity in MS patients and inhibition of convulsions in pediatric epilepsy syndromes. CB1 receptor is a very complex protein. For example, regarding its location, it is usually a presynaptic receptor, which, as you most likely know, mediate the retrograde inhibition of neuronal activity. But the effects of CB1 receptor activation can be different, even opposite, if these receptors are located in excitatory terminals, therefore suppressing excitation, or in inhibitory terminals, therefore suppressing inhibition. The situation is even more complex because CB1 receptor is also located in other neuronal terminals, for example, cholinergic or serotoninergic. It's widely spread in glial cells, and it's also expressed by other cells in the brain, such as vascular endothelial cells, neural progenitors, etc. CB1 receptor cell signal transduction is also very complex itself. CB1 is a very promiscuous receptor that can usually couple to GI proteins, therefore mediating the regulation of a large number of intracellular pathways, but CB1 receptor can also couple to other G proteins such as GQ11 or G1213. Some years ago, we asked whether, at least in part, this complexity and promiscuity of CB1 receptor could be explained by the interaction with still unknown interacting proteins, proteins that could bind to the intracellular domain of the receptor, therefore biasing signaling. And so, 4 years ago, a PhD student in our lab, Carlos Costa, started to tackle this issue. He focused, as I mentioned, in the C-terminal intracellular domain of this metabotropic receptor, specifically amino acids 408 to the terminal 472. He cloned this fragment. He eventually expressed this fragment and conducted 2 different types of high-throughput screening procedures. One is the GIS2 hybrid assays, and Carlos also conducted affinity chromatography-based proteomics. We had a list of several potential interactors. We combined both lists, and we selected 3 different proteins with which we are working now. And today, I will tell you about one of them, which is GRP78, as we will see. For example, in these GIS2 hybrid assays, Carlos cloned the C-terminal domain of CB1 receptor and used it as a bait, exposed to a large collection of human clones as prey. And as you can see here, this blue positive colony, this is very clearly positive, corresponded precisely to the C-terminal domain of this protein, GRP78. You know well GRP78, glucose-regulated protein of 78 kilodalton, also called BIP or ASPA5. I will call it BIP from now on because it's shorter and much easier. as a master regulator of protein folding in the ER, and of course also as a consequence of the unfolded protein response. VIP has been assigned to control many different important issues within the cells, and the classical activity of VIP, the chaperone ATP-assisted activity, is known to rely on the large N-terminal and central domains of the protein. However, the clone of VIP that we found that interacted with CB1 receptor, that we will call from now on VIP CB1 receptor interacting region VIP ER, corresponds to the very C-terminal region of the protein, which suggests that the interaction of VIP with CB1 receptor has nothing to do with the chaperone activity of VIP. Well, the first thing that Carlos did was, of course, trying to validate this interaction by several techniques. For example, he purified CB1 receptor, its C-terminal domain, he purified VIP, he purified VIP ER, and conducted fluorescent polarization protein-protein interaction assays in vitro. And he found that indeed CB1 receptor, C-terminal domain, interacts specifically with both VIP, full-length protein, and VIP IR with a high affinity. In just as other techniques that he used at that time, in HEC293 T cells, he found that CB1 receptor was able to co-IP with full-length VIP and VIP IR. Using bread assays, we also found a specific binding of CB1 receptor C-terminal domain to VIP IR that was displaced as suspected by cold forms of VIP IR or VIP. Using PLA, we also find an interaction between VIP and CB1 receptor, as well as VIP IR and CB1 receptor. And we also asked whether this interaction of VIP with CB1 receptor was able to modulate CB1 receptor signaling. As a first procedure, first technique, we use global assessment of signaling by dynamic mass redistribution assays. As you can see here, when we added a CB1 receptor agonist to HEC cells that express CB1 receptors, as expressed, we found a global increase in signaling. However, this increase in signaling was not evident when VIP, full-length protein, or VIP IR were co-expressed with CB1 receptor. And we didn't find an effect of VIP when using a form of the protein that lacked the interacting region. So we had in hand, at least in vitro, a new interactor of CB1 receptor that inhibited CB1 receptor signaling. And here is when we asked for money to CIBERNED to continue this project. We got we're very lucky, we had the money. And then the four groups, Silvia and Lupe in Barcelona, and Javier and myself in Madrid, started to characterize this interaction both in vitro and in vivo, as we will see later on. Well, with this CIBERNED project, we started to get more in detail about the cell signaling properties of these CB1 receptor VIP complexes. And as I said before, CB1 receptor is an inhibitory metabotropic receptor. It's traditionally coupled to GIO proteins, through which, as you can imagine, it can control cyclic AMP signaling. However, we didn't find any effect of VIP in the coupling of CB1 receptor to different members of GIO proteins, and when measuring cyclic AMP, the main readout of this signaling pathway, we didn't see that VIP exerted any type of effect either on basal forskolin adenylyl cyclase activation or in the Wnt-induced decrease in forskolin activity via, of course, these GIO proteins. So it seems that VIP does not affect the classical inhibitory action of CB1 receptors on the cyclic AMP pathway. We did a phosphoprotein array in which we exposed the cells to vehicle or to WIN and transfected our cells either with control vector or with VIP IR. And we found that there were many proteins of the AKT, mTORC1, and ERK pathways that were affected by WIN and blocked. This increase induced by WIN was blocked by VPR. So what could be the G proteins that could be in mediating this action, we assess the coupling of CB1 receptor to non-GIO proteins, and we found that there was a population of CB1 receptors that were coupled to GQ11, and this coupling was displaced by BPR. In fact, CB1 receptor activation is able, for instance, to increase ERK activation as measured by phosphorylation of ERK. And as you can see here, this activation is attenuated by a GQ pharmacological inhibitor as well as a GQ dominant negative construct. So it seems that BIP affects selectively the GQ11 mediated activation of ERK and AKT mTORC1 signaling by CB1 receptors. What about the brain? First, location. We were aware that BIP is usually ascribed to the endoplasmic reticulum, where our CB1 receptor is located in the plasma membrane with its C-terminal domain facing to the cytosol. So the first thing we wanted to see was whether BIP was also present in the cytoplasm, as it was suggested, in fact, by some previous reports. So indeed, we did cell fractionation experiments, and we found that there was a significant amount of BIP that was present, of course, not only in the ER, but also in the cytosol, in the hippocampus, in the striatum, and in the cortex from mice. So BIP is present in the cytosol, so it's at least plausible that it can interact with the C-terminal domain of CB1 receptor. BIP mRNA was found to be very abundant in the mouse brain, and it was selectively expressed in GABAergic neurons. For instance, you can see here in the CA3 of the hippocampus that the BIP positive cells were also positive for GAD mRNA, whereas the BIP positive cells were not positive for VII-1. So BIP mRNA is mostly located in GABAergic interneurons. We also studied by PLA where CB1 receptor BIP protein complexes were located. And we found that they were also, as suspected, present in GABAergic interneurons. We did assays with many different types of genetically modified models of CB1 receptor. For example, you can see here that control CB1 receptor flux mice express a significant number of cells with PLA positive signal. CB1 full knockout and the conditional knockout of the GABAergic lineage provided a decrease in the signal, whereas animals in which we had deleted CB1 receptor, specifically in the glutamatergic lineage, had the same amount of complexes as control mice. Likewise, we use expression rescue models. Here you see a flux-stop CB1 receptor mouse that is a kind of knockout. We can rescue the PLA signal of the CB1 receptor BIP complexes, either with a systemically expressed CRE recombinase or with a GABAergic lineage selectively expressed CRE recombinase, but we cannot rescue the PLA signal with a glutamatergic lineage selectively expressed CRE recombinase. So CB1 receptor BIP complexes are selectively expressed in GABAergic interneurons and not in excitatory neurons, and we had very similar data in the striatum and the cortex. What about functionality? So we have CB1 receptors coupled to a new protein selectively in GABAergic neurons. The first thing we did was experiments on animal behavior, and we based on the current knowledge that supports that different effects of cannabinoids rely on the activation not of all CB1 receptors in the brain, but on CB1 receptor molecules located on precise neuronal populations. Just to mention a few examples that I will deal with later on. The very well-known hypolocomotor activity of cannabinoids is nowadays known to rely on the selective activation of CB1 receptors located on excitatory neurons of the forebrain. Only those neurons, not others. The well-known cataleptic action of cannabinoids is nowadays known to rely on the selective activation of CB1 receptors located on medium spiny neurons of the direct pathway containing D1 receptors. And the anxiogenic action of high doses of cannabinoids is nowadays known to rely selectively on the activation of CB1 receptors located on GABAergic interneurons of the hippocampus amygdala circuitry. So we had CB1 receptor complexes in GABAergic neurons, so we expected that only behaviors affected by cannabinoids in GABAergic neurons would rely on these complexes. So we did experiments with wild-type mice and mice with a deletion, a partial deletion of VIP, particularly one allele of the two alleles of VIP was deleted. I have to say that VIP minus neural mice are embryonically lethal, so we cannot work with them. So we removed half of the VIP burden in these mice. We injected the mice either with vehicle or a cannabinoid, the main bioactive component of cannabis, THC, and we did a series of behavioral tests. What happened, for instance, when measuring locomotion in the open field test? This test, as I told you, depends on excitatory neurons. So when we measure the ambulation of the animals in the open field test, we found that indeed, as suspected, THC decreased ambulation, but this decrease was similar in VIP wild-type mice and in VIP heterozygous mice, because this behavior relies on glutamatergic neurons, not in GABAergic neurons, where the complexes are supposed to be. What happens with catalepsy? Again, when measuring catalepsy, for instance, with a horizontal bar and measuring the latency of the animals to move, we found that THC exerted, as suspected, its classical cataleptic action, but this action was similar in wild-type and heterozygous mice. What happens with anxiety? That relies on the activation of CB1 receptors on GABAergic interneurons. And here is, as suspected, as we found the effect. THC exerted in the elevated plasmase test, exerted a trend to an anxiogenic effect, and this trend was much more magnified. It was very significant in VIP heterozygous mice, as we expected, because if VIP inhibits CB1 receptor in the VIP heterozygous mice, we should have a higher activity of CB1 receptor, that is, a higher response to THC. That was what we actually found. So as a summary, CB1 receptor is largely coupled to inhibitory GIO proteins through which it decreases the cyclic AMP pathway. That is the classical view of CB1 receptor signaling. We found that CB1 receptor, a population of receptors, is also coupled to GQ proteins through which they mediate an activation of ERK and AKT mTORC1 pathways. And here is when VIP works. VIP can selectively displace GQ11 proteins and therefore block the cannabinoid receptor evoked activation of ERK and AKT mTORC1. We found that CB1 receptor VIP complexes are selectively expressed in the mouse brain in GABAergic neurons, and these complexes control behaviors that are dependent on GABAergic neurons, precisely anxiety. Well, these are the first series of results that we have obtained with the consortium. It was the first part of the project, and we have had this data recently accepted in J Neuroscience. We tried higher, we couldn't make it, doesn't happen, but we are happy with this J Neuroscience paper, in which we, the members of the consortium, have characterized a new interactor of the CB1 receptor protein that, as I showed before, modulates signaling of cannabinoid receptors in the mouse brain, specifically in GABAergic interneurons. Just a few more words, because the second part of the project, and you can also ask, how about neuroprotection? We are in CIBERNED, we are dealing with neuroprotection, of course, and in fact, in the title of the project, we include in cannabinoid, not only activity, but cannabinoid neuroprotective activity. Well, I'm not the kind of guy who likes to put the blame on external circumstances, but here I have to say that we have been very unlucky with the animal models. First, because of coronavirus, of the pandemics, and also because in 3 out of the 4 labs of the consortium, Silvia, Javier, and ours, we have lost colonies. We have had to rederive the colonies, so we have been losing some time very painstaking work to get back to working with the animal models. But now we have taken on the situation and we are working very hard on this part of the project. And now we have already the models. We have some preliminary data that I will show you. And I hope that in the coming months, we'll have more concluding data. We have focused on two different models in which we knew before, of course, that CB1 receptor was neuroprotective. And of course, we had some previous experience with cannabinoids in these models, Huntington disease model and ALS model. Huntington disease model is essentially coordinated by Silvia Ginés in Barcelona. They had cross transgenic mice expressing a fragment of human mutant Huntington exon 1, a pathogenic fragment of mutant Huntington. They have crossed these mice with B plus minus mice. Therefore, we have obtained HD-like animals that lack one of the two BIP alleles, plus, of course, the control genotypes. And regarding ALS, that is coordinated by Javier in Madrid, they have crossed mutant SOD1 mice with B plus minus mice to generate ALS-like mice lacking one BIP allele, plus, of course, the control genotypes. Our hypothesis is, of course, that CB1 receptor is neuroprotective in these models. BIP, as we have shown before, inhibits CB1 receptors, so B plus minus mice will be kind of neuroprotective. So just as an example, time is short and I don't want to make it very long, with the data that Silvia has obtained in Barcelona with the HD model regarding hallmarks of striatal pathology. As you can see here, the transgenic mice, R61, fell very easily from rod to rod, and when one of the two BIP alleles is deleted, these double mutant mice are protected from falling from the rod to rod. And the loss of dendritic spines in the dorsolateral striatum that is characteristic of these HD-like mice is prevented upon removal of one of the BIP alleles. So it seems that indeed BIP is protecting this, and BIP deletion is protecting these animals. Likewise, regarding markers of hippocampal pathology, for instance, these HD-like mice, RC1 mice, are known to have problems in recognition memory, as measured, for example, by the novel object recognition test. And as you can see here, this loss of recognition memory is prevented by deleting one of the BIP alleles in the animals. And as I showed before, for the striatum, the loss of the dendritic spines in CA1, the hippocampus, that is characteristic of these mice, is also prevented by deleting one of the two BIP alleles. So it seems that indeed BIP deletion is protecting these animals, and now we are studying the mechanism, and hopefully in the coming months we'll have more definite results. So here is the consortium again. I think the main strength of the consortium have been putting together their efforts, 2 labs in Barcelona, 2 labs in Madrid, to characterize a new interactor of CB1 receptor with potential implications in experimental, in neurodegeneration, at least so far, in experimental neurodegeneration. And in the name of the 4 PIs of this consortium, I would like to acknowledge, of course, first all the members of our labs that have participated more directly in this project, in this collaborative project. It has been a real pleasure to work with it, and we are still dealing with it. We have to finish up the HD and the ALS data. Hopefully, we'll have 2 papers more in this from our work, one for each of the neurodegenerative diseases. We'd like, of course, to thank CIBERNED, and not only CIBERNED, as I say, it's a diffuse entity because human communities are made by people, by individuals, and we'll also give a special thank to all the members of the CIBERNED staff, Maria Angeles and her colleagues, who try to make our life easier and happy every single day. Of course, we also thank other members of our labs, other collaborators, and other funding agencies. And thank you all for your attention.